This approach is significant because it allows us to visualize and estimate carbon and energy flows from prokaryotes to higher trophic levels in microbial food webs at a very high resolution. This is the only technique currently available for determining which protospecies consume bacteria in situ, and at the same time quantifying the rate of this process at a single-cell level.
Since bacteria are the only significant microbes changing dissolved organic carbon to particulate organic carbon, the knowledge of the players involved and rates at which this happens forms the basis for understanding aquatic ecosystems in general. We highly recommend that researchers become familiar with their study system before they conduct any grazing experiments. This involves light and epifluorescence microscopy, inspection of the samples, and the knowledge of main taxa present and their seasonal dynamics.
Demonstrating the procedure will be Jitka Jezberová, a researcher from our laboratory.
To begin this procedure, collect water samples from the desired location at a suitable depth. Keep the samples in a temperature-controlled cooler, maintained at in situ temperature, during transport to the laboratory. Fix water and trap fluid subsamples for bacterial enumeration with formaldehyde for at least 1 hour to obtain a 2% final volume-to-volume concentration for each sample. First, use a filtration funnel and black filters with a 0.2 micrometer pore size to filter 1 to 2 milliliters of the reservoir water or 10 to 30 microliters of the trap fluid subsample.